Hi everybody, it's Dr. Hinkey here with our last lab presentation. This is on natural selection and the Hardy-Weinberg Principle, where we'll be using those Hardy-Weinberg criteria and equations to determine if a population is evolving or if it is in equilibrium, not evolving, not changing over time. So let's take a look here. Our goals or our learning outcomes objectives for this lab. You should be able to write the Hardy-Weinberg equations for allele frequencies and genotype frequencies. So P and Q are our allele frequencies. P squared, Q squared, 2PQ are genotype frequencies. Given a model population, you should be able to use those equations to find the P and Q values as well as determine the genotype frequencies. And then compare those values to future generations of that population to determine if microevolution has occurred. You should be able to list the 5 conditions that must be met for a population to maintain genetic equilibrium. That is, in order to not evolve, for evolution to not happen, we have to meet these 5 criteria, and you should know those. You should be able to list and define evolutionary agents that disrupt genetic equilibrium. So what are things that can lead to evolution or do lead to evolution? These include mutations, gene flow, genetic drift, natural selection, non-random mating, and sexual selection. So non-random mating is sexual selection. That should be, there are 6 things listed here, but sexual selection means mating is non-random. And explain how adaptations and natural selection lead to evolutionary fitness for a species. You should be familiar with the story in the pre-lab about the Galapagos and Ecuador. So make sure you review that. We are going to just kind of go right into what the lab is here. Evolution is a change in a species over time. We can look at microevolution, which is what we'll do with Hardy-Weinberg principles, which are changes in allele frequencies in a population over a relatively short period of time, meaning over only a few generations, looking at just the same population over generations to see if the frequency of the alleles is changing. Macroevolution is the accumulation of all those small genetic changes over time, leading to a larger cumulative impact of genetic changes that lead to major changes in genotypes and ultimately to speciation. Natural selection is one mechanism through which evolution can occur. This is individuals that are best suited to the current environment are better able to survive and reproduce and pass along their successful survival traits. This is evolutionary fitness. We say survival of the fittest. It's the one that's able to survive the best with the most excess energy after taking care of their needs only needed to survive in order to also go out and find a mate and reproduce. That's a high energy activity, and so you have to actually be able to first survive and meet your own needs before you can pass your genes on. So survival as the first step, but just being able to survive doesn't give you enough excess energy to be able to reproduce as well, so you need to meet both of those. So the fittest is actually the one that passes on their genes at the highest rate. So here's an example of a mouse population. This is showing natural selection. And here we have 1, 2, 3, 4, 5, 6 mice in our initial population, and 1 out of the 6 is white. So if everybody is reproducing at their maximum rate, in this case it's 2 offspring, then in the next generation we're still going to have white, but everybody else reproduced at the same rate, so the percentage of the individuals that are white is still the same. So 1 out of 6, 2 out of 12, which reduces 1 out of 6. So no advantage, everybody at the same. So we'll have this allele in the population, but just continuously at this low frequency compared to the whole population. Then something happens. There's some change in the environment, and we see in the next generation, well, the white organisms are still able to reproduce, but these gray ones, not quite as successfully. So those that survived this change in the environment can go on to a subsequent generation that will be able to reproduce. And because there was a disproportionate number of white individuals surviving, so here we went from 2 out of 12 to 2 out of 5. So differential survival led to each one of them now is able to produce at its maximum rate, and we have many more white organisms here, 4 out of 7, and then 3 out of 4. So we've gone from 1 out of 6 to 3 out of 4 individuals. We've changed our frequency. The more common allele present now is for the white one, and so that change in the environment led to selection for white alleles and white phenotypes. So we're selecting the phenotype that depends on the genotype. All right, so our mechanisms of evolution. Mutation is what really is at the underlying factor for much of evolution, because mutation helps to create variation within a population. So here is our original population. We have mutation that gives us some different varieties. Unfavorable mutations, so mutations are random, right? So they have to occur first. They have to be present first. They occur randomly. Some of them are lethal. Those organisms will not survive, not reproduce, and that mutation will not get passed on. So if it's unfavorable, either the organism can't survive or is not as competitive, that mutation will be eliminated. Other mutations will continue to be passed on at that same percentage in the population that it starts at. And then when there's a change in the environment, that all of a sudden this mutation is now beneficial, whereas here it would just be neutral. They were able to survive and pass on their genes at the same rate as everybody else. But suddenly something changes to make this mutation beneficial. So an example be with antibiotic resistance. So here's my bacteria, we get some mutations. This mutation, not so competitive, doesn't get passed on. Here the mutation doesn't help or harm the bacteria. The mutation has to come before the change in environment. If it's not already present, those organisms will be adversely affected and die. So it has to be present first. The change doesn't cause the mutation. Mutations are random. So this is really chance. So here's my bacteria. So this one has the mutation. This one doesn't. They're both equally successful when there is no antibiotic present. Here we add the antibiotic. These are not resistant, and so they die, but these have that mutation. So now they're able to pass on that gene for resistance to future generations. So that's mutation contributes to evolution. Gene flow. So gene flow is the movement of alleles between populations. So individuals from neighboring populations move back and forth, and that continues to introduce these alleles and keep them present in both populations. In this case, even when we're isolated, we have some migration in and out. So we are introducing this dominant allele to this population and this recessive allele here, keeping them in the population. If we don't have that gene flow, we just have this separation, then we'll get that change in allele frequency. So no gene flow, if we completely isolate populations, then we don't continue to have that exchange of alleles. Genetic drift. Genetic drift is when we have a change in allele frequency due to chance, usually some event that isolates a small portion of a population. would be the bottleneck effect. That would be when we have a large population with a lot of genetic variation in it, and then we kill off lots and lots. There are only a few survivors that go on to reproduce, and their genetic makeup is different. The percentage of alleles in that population differs from the original population, and so all subsequent offspring have a limited allele pool to pick from. And so here we see we have red, green, and blue individuals, but through this bottleneck, let's say all those individuals were hunted to extinction except for a blue and a green one. So now the only allele options we have are blue and green here. That's bottleneck. Founder effect is when a small subset of a population that is not representative of the whole population in terms of the alleles they carry is separated out. So through migration, through some sort of isolation, they begin a new population that has a disproportionate allele distribution compared to the original population. Non-random mating, this is sexual selection. Sexual selection means that there is some phenotype that is more desirable for mating. There is selection, so not all individuals have an equally likely chance of mating because individuals pick mates based on certain phenotypes. So non-random mating will lead to whatever those preferred phenotypes are, to the alleles for those being passed on at a higher rate. So in order for evolution to not occur, mating has to be completely random. Every individual has to have an equally likely chance of passing on their genes. So natural selection is the mechanism through which our process of evolution occurs. So there has to be no selection. For evolution to not occur, we have to have no selection. Any individual is equally likely to survive and reproduce as any other, or every individual is equally likely to die. So natural selection is when there is some phenotype that has a better chance of survival. So this is it in a nutshell. Here we have green and orange beetles, and the crows prefer the green beetles. So over time, the green beetles will be eaten preferentially, leaving more orange beetles to survive to reproductive maturity, to reproduce, until eventually we have this change in population from orange being a relatively minor percentage of the population to a greater percentage to complete. So that population has changed over time. So how do we determine if a population is evolving? We look at the allele frequencies. What are the frequencies of the alleles over multiple generations? So they should reach an equilibrium state, meaning the allele frequencies don't change from one generation to the next. The percentage of different genotypes can change. So the percentage of homozygote recessive, homozygote dominant, and heterozygote, those, the p squared, 2pq, and q squared, those can change as long as when I solve for p and q, the p and q are not changing. Right? So for evolution to not happen, I have to meet all 5 of these conditions, not just any one. And this is Hardy-Weinberg Principle, says in order for evolution to not occur, I must have no mutation taking place. So no spontaneous changes to alleles. Mutation happens. It's just going to randomly happen all the time. It happens. And so that one's pretty tough. How do we get no mutations when that's just something that naturally occurs? No mutations. Mating has to occur randomly. There is no sexual selection. Every individual has to have the same chance of passing on their alleles, and every individual has to pass on their alleles at the same rate as every other individual. The size of the population has to be very large. This is for statistical significance to include all of those alleles. So that bottleneck effect, I have a large population, then suddenly it narrows down, it's very small. So only a few individuals' alleles can be represented in future generations. So with small populations, these changes kind of get magnified. So I need to have a large population for no net change in allele frequency to occur. No migration. I can't have genes moving in and out. I can't have different alleles moving in and out of the population, introducing change to the allele frequency. And no natural selection. No phenotype can be favored over any other. Unless I have all 5 of these criteria being met in a population, that population will change over time. So the Hardy-Weinberg Equilibrium equations, these are 2 equations that you should know. You should know what the letters represent and know how to do the calculations. So the first is for allele frequency. What is the frequency of my dominant and my recessive allele in this population? Within a given population, the total number of alleles is 100% of the alleles. So I can either do this as a percentage with whole numbers or with fractions. P plus Q equals 1. So the proportion of P alleles and the proportion of Q alleles has to equal 1. So I have 2 alleles for a trait. All of those alleles in the population give me my allele frequencies. And to determine the genotype frequencies, use the equation P squared plus 2PQ plus Q squared equals 1. And for this, you should know what each of these represents. My P squared are my percent of homozygous dominant individuals. My 2PQ are my percent of heterozygous individuals. And Q squared are the percent of homozygote recessive individuals. So here I'm looking at allele frequency, 2 alleles per individual. Here I'm looking at my genotype frequency. So here, to figure out this, my genotype frequency, these represent individuals. So here to figure out these proportions, my denominator, what I'm going to divide by, is the total number of individuals in a population that are homozygote dominant, heterozygote, or homozygote recessive. Here I'm going to add up all of my alleles. So my denominator here is going to be the number of individuals times 2, because each individual has 2 alleles for a trait. So take a look at some examples of Hardy-Weinberg. Determine the genotype frequencies from the following allele frequencies. So you're given a population that is in Hardy-Weinberg equilibrium, and P, the frequency of the dominant allele, is 0.3. If I know this, I can figure out the frequency, I can figure out the allelic frequency. What's my proportion of the lowercase allele, my recessive allele in this population, that's represented by Q. And I can also figure out my genotype frequencies. So we know P plus Q is 1. Those are my allele frequencies. So if P equals 0.3, then Q has to be 1 minus P, and my Q is 0.7. So this also points out something. A lot of times students just assume, because of that term dominant allele, that it's also the most frequently occurring. That is not necessarily the case, right? It is the one that, if I have my dominant allele present with my recessive allele, the dominant one is the one that will be expressed. It doesn't mean it's the most frequently occurring one. The environment determines which one of these is favored, which one will be selected for. So here we see that Q, my recessive allele, is actually the one that is more common in this population. All right, so from this population, in order to calculate the frequencies of the homozygous dominant genotype, that's P squared, the heterozygote genotype, 2PQ, and the homozygous recessive genotype, Q squared, well, I just have to put my P and my Q into the genotype frequency equation, which is P squared plus 2PQ plus Q squared equals 1. These are my genotype frequencies. You should remember how to use this equation. This is looking at individuals with this genotype. A genotype has 2 alleles, right? So each individual has 2 alleles. So here, let's look at this example. P is 0.3, Q is 0.7. Let's say that capital letter A is my dominant allele and lowercase a is my recessive allele. How are we going to calculate the frequency of each of the genotypes? Well, all right, my genotypes are homozygous dominant, heterozygote, and homozygote recessive. I have my P and my Q, and P squared gives me the proportion of the population that is homozygote dominant. So 0.3 squared gives me 0.09 or 9% of the population that has the homozygote dominant trait. The heterozygote is represented by 2PQ, can be calculated by this. My heterozygote, if I plug in my allele frequencies, is 0.42 or 42% of my population. And then my final homozygote recessive, Q squared, is 0.7 squared or 0.49, 49% of the population. And if I add 49 plus 42 plus 9, I get 1. 100% or 0.09 plus 0.42 plus 0.49 is 1. 100% of the population is accounted for because these are my only options for genotype in the population with regard to this trait. So it has to equal the entire population. All right, here's another example where we're going to use genotype frequencies for our calculations. So we're looking at the genotype, the number of individuals with certain genotypes, but we're going to look at the recessive phenotype for this. The reason is if I have a dominant and recessive phenotype, and my dominant allele masks expression of the recessive, then my homozygote dominant and my heterozygote individuals have the same phenotype. They look alike. So I don't know, I can't count the number of individuals to figure out my P squared and my 2PQ. Right? My P squared plus 2PQ would be the total that have the dominant phenotype. But I do know that in order to express the recessive phenotype, I have to have homozygote recessive individuals. So just like when we solved our Punnett square and we did a test cross, the only individuals whose genotype I know for sure are those that have the homozygote recessive phenotype. So I can look at that and start with AA, with my lowercase a, my homozygote recessive individuals, to plug in and work in reverse to solve my Hardy-Weinberg equation. So if we have a population of antelope, they're in Hardy-Weinberg Equilibrium, 36% have the recessive trait of short antlers, where A is long antlers, the capital A dominant, and lowercase a is recessive. So I know my homozygote recessives are 36% of the population. This means that, oops, I think that's cut off on your screen, that lowercase a, homozygote recessive, AA, equals 0.36, or 36% of the population. So Q squared represents in that P squared plus 2PQ plus Q squared. The Q squared represents the proportion of the population that's homozygote recessive. Q squared must be 0.36, 36% of the population. So now I can work this out to figure the allelic frequencies, my P and Q. Q squared is my homozygote recessive. It's the square root of 0.36, so Q is 0.6. And I know that P plus Q equals 1 to account for all of the alleles in the population, so P must be 0.4. So now I can plug these back into my genotype frequency calculations and figure out the rest of my population. What portion are homozygote dominant and which are heterozygote. So my homozygote dominant genotype is represented by P squared, and I plug that in and I see that 0.16 or 16% of my population are homozygote dominant. Heterozygote, capital A, lowercase a, is represented by 2PQ, and I have my P and my Q, so I can plug those in, and I see 0.48% or 48% of my population is homozygote or is heterozygote. And Q squared, I know Q was 0.6, 36. Double check, yep, check, that works. Which is what we started out with, so we should end up with the same number, and then I add those up, and 0.36 plus 0.48 plus 0.16 is going to give me 100% of my population. And here's another example. We're going to determine the allele frequencies based on phenotypes and population size. So we have a population in Hardy-Weinberg Equilibrium that contains 50 birds. 38 of these 50 have brown feathers and 12 have blue feathers. Our brown feather allele, capital B, is dominant over the lowercase b that gives me blue feathers in the homozygote recessive. So here's my population. As before, the only the only individuals whose genotype we know for sure are the homozygote recessive birds. So we use this as our starting point. This gave us the number of individuals, so this is giving us that Q squared number. 12 out of my total of 50 are homozygote recessive. So I can use this 12 out of 50, or 0.24, are genotype heterozygote recessive, or BB, 24%. So now I can figure out what that's my Q squared. So what's my Q? We do just what we did in that previous example. So that's my 0.24 becomes my Q squared in this equation. And then what do? I work backwards. Q squared is the homozygote recessive. So I take the square root of 0.24. And I get my Q value as 0.49. P plus Q is 1, so I know my P is 0.51. And then using these figures, I can just plug into the equation to figure out the percentage of my population that is homozygous dominant, heterozygote, and homozygote recessive. Again, just do this as a double check, and if I add 0.5, 0.26, 0.24, it's 1. Right, so how do I know this is, we've been given in all those examples, we're told this population is in equilibrium. But these equations are meant to help us determine, is a population in equilibrium or not? So let's take a look at this as population. Here's my starting population. 3, 4, 5, 6, 7, 8, 9, 10. Oh, I have 11 individuals. There's supposed to be 10. Here's my population. Let's get rid of one of those. Whoops. Let's get rid of one of these so that I'm not showing you something that's not correct. Right, here's my population. 7 black individuals, 3 white individuals. B, the capital B allele, as the black is dominant to white. And so how will I go about calculating my PQ, P squared, 2PQ, Q squared? Right, the total number of alleles in the population is 2 times the number of individuals. I have 10 individuals, so I have 20 alleles. I don't know if the black individuals that I see here are homozygote dominant or heterozygote, but I do know the white individuals are homozygote recessive, so have 2 small b's. So I can calculate Q. Q is the number of white individuals times 2, because each one has 2 lowercase b alleles, divided by the total alleles in the population. If there are 10 individuals, there must be 20 alleles, which is 6 over 20 or 0.3. And then I know P plus Q is 1. So 1 minus Q is P. My P is 0.7. I could also calculate P squared 2PQ and Q squared for this population. So P squared is a percent that is homozygous dominant, 2PQ the percent that's heterozygote, Q squared the percent that are homozygote recessive. Again, I don't know if these black individuals are homozygote dominant or if they are heterozygote, so I'll start with those white individuals. And I can calculate those. Q squared, 3 squared, 0.09. And then, well, I could actually just plug in. I already figured out my Q, so I can plug, or my P, I can plug that in. 0.49, 0.42, 0.09. Now let's look at this population. Whoops, after 5 generations, is this population in Hardy-Weinberg equilibrium? Well, I would calculate those same values, my P and my Q. So here's my Q now. I have 2, 4, 6 individuals. So I have 12 lowercase b alleles out of 24 total. It's 0.5. Those are my totals. And I calculate if this population, this was my starting proportion, this was my starting P and Q, and these are my final P and Q, and I see that my P and Q values have changed, and so the population is not in equilibrium, it's evolving. So that's how I use these formulas. As I compare, I have to have a set, what are my P and Q to begin with, and what are my P and Q after multiple generations. And again, my number of individuals, my P squared, my 2PQ, and my Q squared, those can change, and it doesn't necessarily mean that my P and Q are changing. So if I start with completely homozygote dominant and completely homozygote recessive population and allow random mating over time, they're going to be heterozygotes. But my P and Q, when I calculate my P and my Q, as long as they're the same as that original population, then that population's not evolving. So these genotype frequencies can change over time, and I can still be in equilibrium. It's my P and my Q that determine whether or not I'm in equilibrium. All right, so on to your lab. Go over the PowerPoint, watch this video, do your top hat activities, complete the virtual lab, and your post-lab questions, review the study guide, those learning objectives, and then you will be all set for Lab Practical 2. And we'll have that Lab Practical 2 review coming up in just about an hour, and then you'll be ready for Practical 2 that covers everything from cellular respiration through this lab. All right, thank you all, and good luck on your practical.